So how else might we sort of address this big challenge that we have in terms of, the vast majority of our drugs do not work really well and really early for the vast majority of our patients? Well, the other obvious thing is discovery. And actually, in terms of actually developing new drugs that could work a lot better. But therein, there is also a problem. And that is that if you look back, and these numbers, slightly sadly have not really changed over the past 20, 30 years. If you look at the success of all the drugs going into phase one studies that are intended to be the next big, blockbuster agent in a particular disease, only about 1 in 10 of those drugs actually ever ends up being approved for the disease it was meant to be going into. And you can this is for all autoimmunity. In fact, in gastroenterology, it's even lower. So why is that? Well, the vast majority of these drugs actually fail at phase two. And as many of you will know, the job of a phase two study is just to very simply ask the question, does this drug actually work in some sort of dose for my patients? And the important thing to take away from this is, this big purple half of the pie chart here is efficacy. So when the question is, does this drug actually work for my patients? The simple answer is no. That doesn't mean the drug is not blocking the kinase it's designed to block or mopping up the cytokine it's designed to mop up. It just means that doing that in someone with inflammatory bowel disease or rheumatoid arthritis or ankylosing spondylitis actually doesn't make a blind bit of difference to the disease activity. And so I think we also need to sort of take a step back and think, well, what's that telling us? Because we're in a situation where even the drugs we have only work in a minority of our patients, and our attempts to develop new drugs often fail, more often fail than not. So what is that telling us? Well, I think fundamentally what it's telling us is that we still don't understand these diseases well enough. Because if we did, we wouldn't be targeting the wrong biology, such that the efficacy doesn't work out in terms of developing the drugs, and we would hopefully have drugs that work in more than sort of 20% or so of our patients. So then the natural question, of course, then is how can we better understand these diseases? And I've put IBD here, but I think this is probably applicable to most of the sorts of diseases we're interested in. Now, I'm biased as a geneticist, but I would argue that actually genetics gives us a pretty unique opportunity to better understand these diseases. And that's for a couple of reasons. The first is that genetic discovery has been incredibly successful since the advent of genome-wide association studies. So we now know of hundreds of regions of our DNA that are directly involved in the development of these diseases. The other nice thing about genetics is genetics really nicely resolves the sort of cause-effect relationship. Because if you're looking in the microbiome or the metabolome or the transcriptome, it's always difficult to unpick whether or not the changes you're seeing were a cause of the disease, or maybe they were caused by the disease or the treatment that the patient's got. Whereas genetics always comes first because your DNA is set when you're born. So if there are genetic associations, we know that this can't be caused by the disease, it must be contributing to the disease process. And actually, consistent with that, if you take those drugs going into phase one studies and you look at the ones that are targeting pathways implicated by genetics, they actually have a far higher chance of working. Now, that was originally estimated to be maybe a twofold higher chance, but actually, if you really understand the pathway, it's probably up around a six or sevenfold higher chance. So you could take that 10% success rate and turn it into something like a 60 or 70% success rate if only you know exactly what the right pathway is to target. But the problem we have is that the majority of these genetic associations do not lie in the coding sequence of genes. So they're not having predictable effects on protein structure or function. In fact, the vast majority of these associations lie in the non-coding DNA, and they're very strongly enriched within these things called enhancers. And enhancers are the sort of control elements that determine whether genes are highly expressed or not at all expressed within particular cells. Remember, every cell in your body has the same DNA, but what makes a hepatocyte look like a hepatocyte and a neuron look like a neuron are the genes that are turned on in those particular cells. And the thing that controls that is your enhancers. But the problem is that enhancers often lie quite a long way away from the genes they regulate. They sometimes only work in specific cell types. So in order to then try and figure out the biology, you've first got to identify the right cell type. You've then got to figure out what the gene that's being regulated is. And only then can you start thinking about how the genetics might be perturbing this. And what all of that has meant is that while there's an amazing potential for genetics to tell us more about these diseases, actually at the moment, most of that potential is largely unrealized. So I want to just finish by telling you a story that we published last year. And this was interestingly looking at a region on chromosome 21 that's a gene desert. Gene desert just simply means there are no obvious genes at the site of the association. And the interesting thing is that this is a gene desert that's not just linked to inflammatory bowel disease, it actually predisposes to at least 5 different inflammatory diseases. And we've known of all of these associations for well over 10 years. It's just that because this lies in a gene desert and we can't make up a nice story about it when we write our genetics papers, this is one of those regions that's been largely ignored. So we reasoned that this must contain one of these enhancers, one of these control elements, and it must be an enhancer that was functioning in an immune cell because that was the common link between all of these different diseases. And what we ended up showing, the methods we used it for here doesn't matter, but essentially we showed that it was an enhancer and it was only being switched on in monocytes and macrophages. And while there were some nearby genes, these ones here, which people had assumed might be involved, in fact, what we showed was that it wasn't either of these genes that was the target of this enhancer. It was actually a gene that was about 300 kilobases away, so really quite a long way away. And the way this works is, although the DNA, I've presented it here as a nice straight line, actually, if you look inside a nucleus, it's much more like a bowl of cooked spaghetti, where there's bits over this side which are physically touching bits over here by looping things together. The other thing we realized was that actually, if you carry the risk allele, you tended to have higher levels of this gene, ETS2, in your monocytes and macrophages. So the genetics here is very clearly pointing at a specific gene, and it's very clearly pointing at a specific gene within a specific cell type. But no one really knew what ETS2 did in monocytes and macrophages. Um, One of the things we do in the lab is we are able to play around with cells. We can take primary immune cells, we can use CRISPR to either knock in or knock out genes. And what we did as a very simple experiment to begin with was we made inflammatory macrophages, but we used CRISPR just to delete the ETS2 gene. So we effectively turned it off. And what we found was that there was a large number of genes that changed in terms of their expression. And lots of these were genes that are known sort of initiators or amplifiers of inflammation. So you've got chemokines, you've got cytokines, you've got signaling molecules, you've got pattern recognition molecules, you've got some secreted effector molecules. And in fact, when we did pathway analysis on this, what we found was that lots of different macrophage inflammatory processes all seem to go down when you just take away ETS2, whether that's phagocytosis or cytokine production or reactive oxygen species production. But we then remembered that, it's all very well knocking a gene out to figure out what it might do. But in the people who carry the risk allele, they have a bit more ETS2. So maybe, as well as being necessary for all these processes, maybe ETS2 was sufficient to turn them on as well. And there aren't that many examples in biology of when things are both necessary and sufficient. And when you find them, they're often the sort of the key controllers. Often people call them master regulators. And so what we did was a simple experiment where we essentially made resting non-activated macrophages, but we can load them up with mRNA encoding ETS2. So this is exactly the same technology that's used to make your COVID vaccine, where we can get in vitro transcribed ETS2, we can load it into the cells, and then we can begin a very small inflammatory response to see what happens. And strikingly, we found that all of these inflammatory processes turn on, and they turn on in a dose dependent manner. So the more ETS2 you put in, the more these processes all turn on. So what we have now found is a gene that appears to be the sort of the control switch, the sort of the central orchestrator of this process. And actually, really interestingly, if you take the resting macrophages in which you've loaded up ETS2, those macrophages end up looking almost exactly like the macrophages that you can find in the gut of patients with Crohn's disease. You can do that from taking single cell RNA-seq signatures of inflammatory macrophages, and the majority of these genes are turned on when you turn on ETS2. And that includes all of our favorite drug targets, things like IL-23 and TNF and IL-1 beta. So, we've known for some time that macrophage inflammation is critically important in autoimmune and inflammatory diseases. We know this for lots of reasons. We know it because some of our best drugs target the cytokines that are principally produced by these sorts of macrophages. We know it because the macrophages are the ones producing the ROS that damage the tissue, or the matrix metalloproteinases that remodel tissue. And as I said to you earlier, we're starting to realize that putting these drugs together might be better than using them on their own. So far, people have looked at blocking TNF and IL-23. It won't be long before people want to start adding more of these things in. And I think the place we're going to get to is the realization that the best chance we have of getting the patient well is having a broad effect to try and turn down a whole series of different inflammatory cytokines, for example. And you might conceivably do that by giving people 10 monoclonal antibodies, but which healthcare provider is ever going to pay for that? The other way you theoretically could do this is if you could find the thing that switches them on at the first place, the thing that sort of sits at the top of the pyramid. And we think that may be what ETS2 is. So in terms of could this actually provide us a therapeutic opportunity, coming back to the point about, we need better therapies that work in more people. We actually initially looked at this in an in silico way. We took a large number of gene signatures that have been derived from cell lines treated with that one of thousands of different small molecules. So this was about 30,000 different signatures. And using that, we could just simply say, do any of these signatures look like they may be acting by knocking out ETS2 or turning down ETS2? Because we know what that looks like because we've got all of that data. And several of them did. And excitingly, some of these are already drugs that are approved. So, for example, JAK inhibitors. The other thing we found, and this is all the ones in red here, all of these are known upstream regulators of ETS family members. So, we could almost rebuild everything that's known about the regulation of ETS family members from this experiment. And the thing that come out top were MEK inhibitors. And that's really interesting because MEK inhibitors are already licensed for use in people. They're used for cancer, they're used for neurofibromatosis and other conditions. They're not used for anything inflammatory. And so we just did a very simple experiment where we made these inflammatory macrophages, we treated them with a MEK inhibitor, and sure enough, all of the inflammatory pathways seemed to turn off. And in fact, we could take that one step further because we could take biopsies from patients with active inflammatory bowel disease before they received any treatment. And those biopsies can be cultured for up to 24 hours at an air liquid interface. And in that time, they will continue to release all the pro inflammatory cytokines they were releasing when that bit of colon was part of the person. And we can add in drugs into that media. So we did that. We added in a MEK inhibitor. And sure enough, this seems to reduce the release of a whole sort of panel of different pro-inflammatory cytokines, including TNF and IL-23, but also things like IL-1 beta that's been directly linked to treatment resistance in the past. And transcriptionally, this also seems to turn off a large number of genes whose expression reflects the relative amount of inflammation. So this isn't to say we should all be rushing out and using MEK inhibitors in our patients tomorrow. Of course not. We know that MEK inhibitors do have side effects that limit their long-term use in cancer and things like that at the moment. So actually one of the things we're doing now in the lab is trying to work out a way whereby we can use this, but actually make it safe enough to be used for people in the future. But this won't be the only key pathway that's involved. And actually, there's a lot of work now going on in the lab to try and understand these other things to see whether or not we can actually come up with sort of rationally designed drugs that are specifically targeting the key nodes within the pathways that are ultimately driving these diseases. So I think the therapeutic ceiling, as it's been referred to, is something that's well known in many different immune-mediated diseases. Although this is ulcerative colitis, I'm sure you could draw similar things for rheumatoid arthritis and multiple sclerosis and so on. And I think it's a really interesting thing to ponder in terms of how are we going to get out of this? And it may be a combination, both in terms of precision medicine and trying to use the drugs we already have in a better way, but potentially also in terms of thinking about discovery science, and genetics is just one way of doing this, in terms of finding better targets so that the next generation of therapies won't just work in 20% of people, but hopefully will work in some more.